Good morning, students. In this class, I am going to discuss with you about Rhizopus sexual reproduction. Sexual reproduction in case of Rhizopus takes place by gametangial copulation. During sexual reproduction, two morphologically similar but physiologically different hyphae are produced. The two hyphae belongs to two different strains, that is positive and negative strain. This phenomena is called as heterothallism. Sexual reproduction in Rhizopus takes place by the gametangial copulation. The gametangia are morphologically similar but physiologically different in nature. The two hyphae belongs to the two different strains, that is one belongs to the positive strain and other belongs to the negative strain. The fusion occur between these two gametangia and they are produced in different thallus. This phenomena is called as the heterothallism. Hetero refers to different, thallism refers to thallus. So, because of the presence of two types of thallus involved in the reproduction, this condition is called as heterothallism. During sexual process, two compatible hyphae come nearer to each other. Of the two hyphae, one belongs to the positive strain and the other belongs to the negative strain. Each hyphae produces a branch called as the copulating branch, and this branch is called as progametangium. This progametangium resulted in the formation of terminal gametangia, and the remaining part is considered as suspensor. As the gametangia are multinucleated, this condition of gametangia is called as xenogametangia. The two xenogametangia united resulted in the formation of the diploid structure called as the zygospore. If the two gametangia are similar in nature, then that condition is called as isogamy. If the two gametangia are dissimilar in nature, then the fusion is called as the anisogamy. During sexual process, two compatible hyphae come close to each other. These two hyphae, one belongs to the positive strain and the other belongs to the negative strain. Each hyphae starts to produce the copulating branch, and this copulating branch is considered as progametangium. This gametangia is differentiated into two regions. One is gametangium and the rest part as suspensa. Because of the presence of multinucleus in the gametangia, it is called as xenogametangia. Fusion of these two xenogametangia resulted in the formation of the spore called as the zygospore. If the fusion takes place between the two same hyphae, then it is called as isogamy. If the fusion takes place between the two gametangia which are dissimilar in structure, then that fusion is considered as the anisogamy. Zygospore. The zygospore in Rhizopus is usually spherical in structure with spiny in outgrowth. The wall of the zygospore is differentiated into two regions, that is outer exine and inner intine. The zygospore has many diploid nuclei in it. During germination, zygospore produces the short mycelium, and that mycelium is called as the promycelium. The tip of the promycelium starts to enlarges and it forms a structure called as the zygosporangium. Zygospore in case of the Rhizopus is formed by the fusion of two gametangia, and it is spherical structure, spiny in nature. It is having two wall layer. Outer wall layer is called as the exine and the inner wall layer is called as the intine. This zygospore is diploid in condition and it produces a short mycelium called as the promycelium. This promycelium starts to enlarges in its size at its tip and it is called as the zygosporangium. The diploid nuclei in the zygosporangium undergo the continuous meiotic and the repeated mitotic division, which resulted in the formation of 50% of positive strain and 50% of negative strain. The spores of the positive strain germinate to produce positive hyphae, and the spores of the negative strain produces the negative hyphae. The diploid nuclei undergo the meiotic division and the repeated mitotic division, which resulted in the formation of 50% of positive spores, which belongs to the positive strain, and 50% of negative spores belongs to the negative strain. These strains later germinate to produce the new hyphae. Positive strain produces the positive hyphae. Similarly, negative strain produces the negative hyphae. This is about the sexual reproduction in case of the Rhizopus. Let us move towards the next topic, that is the life cycle. The hyphae of Rhizopus undergo two types of reproduction. One is the asexual reproduction, another one is the sexual reproduction. In case of the asexual reproduction, hyphae started to produce the stalk-like structure, which is called as sporangiophore. At the tip of the sporangiophore, it produces the sac-like structure is called as the sporangium. Within the sporangium, the spores are produced is called as the sporangiospores. When this sporangiospore starts to germinate, it produces the new hyphae. Similarly, in case of sexual reproduction, the mycelia produces two types of hyphae. The one is considered as positive hyphae, another one is considered as negative hyphae. Both are morphologically similar in nature, but they are physiologically differ in nature. The positive hyphae produces the positive gametangia. Similarly, negative hyphae produces the negative gametangia. These gametangia come close to each other and they starts to copulate and produces a structure which is diploid in nature called as the zygospore. This zygospore produces a promycelium which starts to swell and form a sac-like structure called as the zygosporangium. This zygosporangium is diploid in condition, which undergo continuous meiotic and mitotic division and resulted in the formation of 50% of positive spores and the 50% of negative spores. Positive spores produces the positive hyphae and negative spores produces the negative hyphae. This is about the life cycle taking place in case of the Rhizopus. Thank you.